Hello everyone, I am Sumit Gunjal, student of the Elite Institute of Pharma Skills, Pune. Today, I am having the demonstration on clinical trial design. What actually the work is done in clinical trial design, we are going to see in today's demonstration. And before that, we will know what is clinical trial, what actual work is done in clinical trial design, how it is formatted. Let's have a detailed look. Clinical trial, it is a systematic study when a new research is done on a subject human, then we come to know what actual effect is, what will be the adverse effect of new drug. And the aim, motto of clinical trial design is to determine the safety and efficacy of the new drug, and our actual subject, that is clinical trial design, it is defined as the structure, the sequence or the structure of the activities which are going to carry during the clinical trial. It is known as clinical trial design. What actual work we are going to do and in which sequence we are going to do, it is clinical trial design. The clinical research study is categorized into two groups, that is observational study and experimental study. Observational study, it is either descriptive or analytical, and experimental study is completely experimental, but it consists of random allocation. Let's see in detail observational study. The observational study is to evaluate the accuracy, whatever test procedure, test, or whatever measures we are taking, we have to look over that, how it is carried out. The observation is done by investigator, but by doing so, we come to know the strength and circumstances of the observed. It is also of two types, that is prospective study and retrospective study. Prospective study, it includes what will happen and what are the future outcomes. It is divided into three types: concurrent study, non-concurrent study, and cross-sectional study. Concurrent study, whether the characteristics are risky or protective, it is determined here. Non-concurrent study, the information except which is exposed and which is not exposed is taken under the consideration of the present. Cross-sectional study, at one single point we come to know and we have to do the comparison of the persons who were included in the clinical trial and what actually the characteristics are done, what actual effect is shown, or either the effect is not shown, both are compared. It is cross-sectional study. Next is retrospective study. It determines that what had happened by looking the previous condition is taken under the consideration. It is of two type: retrospective study or cross sectional retrospective study. Retrospective study, all the characteristics are exposed for the longer time, and after that it is considered that the effect shown by the person is considered to as a case, and the persons who are not showing the effect, they are considered as a control, so it is called as an case controlled study. Next is cross-sectional retrospective study. It is also called as an transversal retrospective study. In this study, the exposure is sought only in the present. Next is experimental study. All experimental work is carried out and all the influence investigator are exposed to this subject. The main motto of this is to demonstrate the safety and efficacy of the new drug. It is also classified into two trials, that is community trial and clinical trial. Community trial involves the complete community interventions and it is done on all. Clinical trial, as we have seen the definition at the starting of the demonstration, it is same, and it is completely research oriented study. Now we will start to our actual format, that is what is the categorization of clinical trial design. It is categorized into five groups, that is treatment trial, prevention trial, diagnostic trial, screening trial, and quality of life. In this treatment trial, we actually come to know which treatment should be given. That is, it might be new combinations of two drugs, it might be new approach to surgery, it might be a new therapy which helps to boost the immune system and fight with the disease. Prevention trial, all preventive measures are taken under the consideration, but it includes healthy peoples, that is, it might include vaccines, minerals, medicines, vitamins, and lifestyle changes. Diagnostic trial, only the diagnosis and the condition to detect the disease is done. Screening trial, the screening of the disease should be done, means very early we should come to know, or we should find out the what disease is there. If the screening trial is effective, it will reduce the number of dead particulars disease. Next is quality of life. How we can improve the quality of the life, how we can improve the people who are with the pain, sleep disorder, nausea, nutrition problem, depression, etc. How we can give them a comfort life or quality life. All this is seen under the quality of the life. Next is types of clinical trial design. There are various types of clinical trial design, in which first is parallel design, second is crossover design, third is factorial design. First we'll see parallel design, the work or the design which carries out the carry in a parallel way, but the treatments are not mixed. It is called as a parallel design. We will see one flow chart. Here we are seeing group A, group B. There are two groups and we are giving them separate treatment. Group A will receive treatment A, group B will receive treatment B. All this is done randomly. The treatments are given randomly. Any treatment should be given to any group, but it should be throughout, it should be same throughout the clinical trial. And after that we will know what are the outcomes, what evaluation and reports is done. Next is crossover design. Word itself indicate that crossing is done, means the treatments are alternatively done. More than one treatment is given to a group. That is, if we consider group A, group B, and if we give them a treatment, that is group A given treatment A, group B given treatment B, randomly for first week this treatment is carried out. But as we enter in the second week, the treatments are exchanged. That is, group A will receive treatment B and group B will receive treatment A, and after that data is analyzed. Next is factorial design. In factors, the word itself indicate that the all the design is in factor. If we consider four groups, that is group A, B, C, and D, and the treatments are given in such a manner that group A will receive treatment A, group B will receive treatment B, group C will receive the combination of treatment A plus B, and group D will receive no treatment, that is only placebo drug is given. And this is in factor, so why it is called as factorial design, and at the end we do the evaluation. So these are the types of clinical designs. Next is bias. When we consider it, when we consider the bias, it means that the epidemiological or distortion fundamental concept in epidemiology, it is called bias. It is error. It might be in collection, it might be during analysis, it might be during interpretation, it might be during publication, it might be at any time, or it might arise at various stages in the clinical trial. It is of different types, that is selection bias, performance bias, it may be detection bias, it may be attrition bias, or it may be publication bias. Mm, okay, and to ensure that there is no bias, we have to do randomization, blinding, control group, or sample size. These four methods can decrease the randomization. Next, we will see one by one. Randomization. What is randomization? It is without any plan. Random means without any plan. Whatever we are doing, it will be without any plan. Okay, randomization means the process of assigning the treatment to the participants or the group for the study of clinical trial and giving the equal chances to the groups and participant. It is called randomization. For example, if there is a participant, we are selecting them randomly, means randomization is carried out. There are two groups. First is control group, second is investigational group. Or we can say that the control group is given standard treatment and the investigational group is given new treatment. But in between this, we have to take a follow-up what actually is happening, and then we are going to take outcomes. The basic benefits of randomization: it eliminates the bias. First and most important, that it eliminates the bias. It maintains the balance between prognostic variables and the in general, if we do randomization, it is the essential tool efficacy to show the efficacy of the treatment. Next is blinding. Blinding means if uh, if if the person who is taking the treatment, he does not know which treatment he is taking, then it may be blinding. If the person who is giving the treatment, it he doesn't know which treatment he is giving, it is a blinding. And this is called blinding, where participant, investigator, or assessor, participant is the person who is taking part in clinical trial, and investigator is who is healthcare provider. Statistician means person who collect the data. They all are unaware of the treatment. That is called blinding. And the blinding can be done in following ways. First is single blinding or single blind, where patient is only blind, doctor know which treatment he is giving, but patient does not know which treatment he is pursuing. So this comes under the single blind. Second is double blind, where doctor and patient both are blind, both doesn't know what they are doing. Doctor doesn't know which treatment he is giving to patient, and patient also doesn't know which treatment he is taking. So this comes under the double blind. Only statistician know which treatment, which experiment is going on. Next is triple blind, where all three of them are blind, means participant, investigator, and statistician all are blind throughout the trial. They doesn't know which what is actual work going on. Next is control group. In the picture you can see that what actually the control group is and what actually the experimental group is. We can say that control group is a baseline group and it is a comparison group, where we are doing the comparison. In control group we have to compare our treatment with the known treatment, means unknown treatment is compare with the standard treatment, and the control group is exactly in the same way, and the treatment which is given to control group is exactly in the same way which is given to the experimental group, and participant does not know that whether he she is in control group. The participant doesn't know in which group he is, and it is decided by randomization. Types of control group. First is placebo concurrent control, where experimental group receives the standard treatment, but control group receives the placebo treatment. Second is no treatment concurrent, means experimental group receives the treatment, but control group does not receive any treatment. Third is dose response concurrent, where the comparison of the dose is done, means two different doses are administered and which are different for each other, and then they are compared. Fourth is active concurrent, where experimental group is given treatment but control group is given therapy to know which therapy is effective. And fifth is historical control, where only experimental group is given treatment but the control group is compiled from his historical data. Next is sample size. Number of participants or the observation includes in the clinical trial study is called an sample size. The subject should be large enough to provide the reliable answers to the question. That is, the persons or the groups who are taking part in the clinical trial, they are called as in participants, and the total number of them in collection, it is called as an sample size. And they should be in large quantity, large enough to give the proper answers to the questions which will be arise after clinical trial, and it is denoted by n. There are two effect two two properties which affect the sample size, that is precision and study to draw the conclusion. And this is all about the clinical trial design. In this way, the clinical trial design is formatted, and this is how the clinical trials are carried out. So from this demonstration, at the end we come to know that we should follow these rules and regulations. We should follow the randomization, we should follow blinding, we should follow sample size, we should follow control group, we should do three of designs, that is parallel design, crossover design, factorial design, to avoid the bias and to give a proper effective treatment at the end. And this is in, if we work in this way, I hope that our research will grow, our research will be 100% successful. Thank you so much.